the rest of the double helix from getting all messed up and torquey. And so helicase is here unwinding the helix. You've got your single-stranded binding proteins right here. And then you have your primase, which is making an RNA primer. And that RNA primer is going to serve as the landing pad for DNA polymerase III. And so DNA polymerase III, if you look at the molecular level, remember the template, or the original piece, is in the dark blue, and the new DNA is in the light blue. So when DNA polymerase comes in, how's it actually adding nucleotides? And so the answer is that it's using a type of molecules called DNTPs. These are deoxyribonucleotide triphosphates. So DNA is deoxyribonucleic acid. This is deoxyribonucleotide triphosphates. So ignore this, and in place of this, just put in GTP, ATP, TTP, or CTP. All right, that's what we're talking about. So it's still a molecule, it's still a nucleotide, except it's got 3 phosphates attached to it instead of just 1. And so, and I might as well write this out for you. Let's see, ribonucleotide triphosphate. DNTPs. And so, as we said, there can be 4 different DNTPs because we're building DNA. So you can have GTP, ATP, TTP, or CTP come in. And depending on which molecule is being brought in, so in this case, DNA polymerase is looking at the template strand and it sees an A. So it knows it needs to put in a T. So it'll bring in one of these TTP molecules. All right, you still have the nitrogenous base and the sugar and a phosphate. So it's just like a nucleotide, except it's got 3 phosphates instead of 1. And the reason why it has those 3 phosphates is because we're building DNA, right? So it's going to require energy. So we're going to release that energy by hydrolyzing those phosphate groups off. So these DNTPs, they serve 2 purposes. All right, they provide the energy required to build the piece of DNA, and they also provide, the base of the nucleotide that is going to make up the new piece of DNA. And so this TTP will come in, it will, hydrogen bond, the T will hydrogen bond with the A, and then in order to build this new piece of molecule, you're going to end up removing those 2 phosphate groups, releasing energy, and then that leaves you with 1 phosphate group left, which is now going to come and attach at this 3' carbon right here. So you're literally just bringing in the DNTP, you're cutting off a couple of phosphates, and then that 1 remaining phosphate is now going to bind to this 3' end of the new piece of DNA. And so you're just going to continue doing this until you reach the end of your DNA. And so in this case, it's going to do the same thing. After it adds this T right here, it's going to see this G and say, okay, I need to put in a C, it's going to see the C and know that it needs to put in a G. So then a GTP molecule would come in, cleave off the 2 phosphates, attach the phosphate to this, and then keep going on. Okay, so when we're talking about DNA replication, it sounds so simple. It's like, okay, just make a piece of DNA and put it in the opposite nucleotide. Well, the idea behind it is simple, but the actual mechanism or mechanics behind it is actually somewhat complicated. And it's complicated because of this anti-parallel nature of DNA. So I'm going to draw a new replication fork so you can see what I'm talking about right here. So, all right, so here's my replication fork. And here, this piece is 5' to 3'. This piece is 3' to 5'. So when I start making my new DNA, both strands will be used to make DNA. So both strands are being replicated at the same time. And so when I'm making my new piece of DNA, my new piece of DNA is going to be running in this case. If this piece is 5' to 3', that means this piece is going to be 3' to 5'. So in other words, remember we said we can only synthesize DNA in the 5' to 3' direction. So we're going to be adding nucleotides to this end. So as the helicase is moving this way and replication is moving this way, then the new pieces of DNA can just be added, followed right behind the helicase. So it'll just follow behind this whole replication fork and add nucleotides to the 3' end. Pretty straightforward. And so you end up with one long, continuous piece of DNA. Now, this new piece of DNA that we're making, this is called the leading strand. And the leading strand, the leading strand is going to have two characteristics. It's going to be going towards the replication fork. It's going this way. It's following behind the fork. And so towards the replication fork. And then the second characteristic that you need to know is that it's made continuously. In other words, that we have one long piece of DNA. So it's made continuously. Now the other strand, this strand, is going to act a little differently because this strand starts with 3' and ends at 5'. So when we make our new nucleotides, or our new piece of DNA, we're making them 5' to 3'. But replication's going this way. So if I've got all this stuff that's going this way and I can only synthesize this way, that's going to give me issues. And so this new piece of DNA that's going to be made, this is called an Okazaki fragment. And so what's going to happen is you got to remember everything's moving this way. So this was a first initial piece of DNA that was made, and it's just a little chunk of DNA because it can't go this way because now this stuff over here is still double-stranded. So you got this strand being made this way, but everything else going this way. So the way that it fixes this, it will make a little piece of DNA. So DNA polymerase 3 is still here doing the replication. It'll start at one of these RNA primers that are not drawn on here, but imagine there's a primer there, a primer there, a primer there, and then this, of course, would have its initial primer as well. And so when DNA polymerase 3 comes in, it's going to start at that primer and then synthesize the DNA. Now on the lagging strand, which is what this new strand is, the lagging strand, again, you have a primer at various different locations. Unlike the leading strand, which you only need one primer because you're only making one long continuous piece of DNA. For every piece of DNA you make, you have to have a primer. So the lagging strand is going to have multiple primers, while the leading strand is only going to have one primer. Now, so we're synthesizing our DNA, and the way that this is going to happen, because remember, everything's moving this way. So this was going to be the first primer that was put in. This was going to be the second primer that was put in, and this was the third one. So what's going to happen is as this replication fork is moving this way, DNA polymerase is going to come in, jump onto the first primer, synthesize the new piece of DNA, just a short little segment of it, and then what's going to happen, DNA polymerase, he's going to jump backwards and do the same thing. So it's like two steps forward, one step back type of thing. So now he's going to jump from this piece of molecule, he's going to jump over here. And then that's going to synthesize this new piece of DNA until it runs into that next primer. And then again, he's going to jump backwards and go this way. So you're still adding nucleotides to the 3' end. You're just having to do it a little piece at a time instead of this long, continuous piece of DNA. And so the reason why they're acting different is because the directionality of each strand. One's going 5' to 3', which means when you make your new DNA, you're just adding nucleotides to the 3' end behind the replication fork. But on the lagging strand, because you can only add at the 3' end, you have to add a little bit and then jump back, and add a little bit, and then jump back. And so the lagging strand is going to be synthesized. You're still moving with the flow of the replication fork. It's just you're making this DNA in these little bitty pieces of segments. So the leading strand is synthesized towards the replication fork and continuously, while the lagging strand is going to be synthesized away from the replication fork and in segments or in pieces, non-continuously, you could say. And so those fragments that we're making, they actually have a name for the Japanese scientists who discovered them. These guys are called Okazaki fragments. Okazaki fragments. And so we're synthesizing our DNA on both strands. It's just different ways on the leading strand compared to the lagging strand. And so we can see our replication bubble right here. And so for this strand, again, this is going to be 5' to 3'. So when we make our new piece of DNA, we start with one primer at the origin of replication. DNA polymerase 3 will come in and synthesize towards the replication fork, and it'll synthesize a continuous piece of DNA. So that new piece is the leading strand. Now the lagging strand, again, is going to come about because of the directionality of the DNA. This bottom piece is going 3' to 5', so our new pieces of DNA that we're making are going to be added to the 3' end of the new DNA molecule. And so this lagging strand, this is going to have multiple different primers. And so you're going to have DNA polymerase here come in, land on this first primer, synthesize the new DNA. And once it runs into this primer, it's going to jump backwards and then synthesize again until it runs into that primer, and then it's going to jump back, so it's going to continue jumping back and synthesizing. So it's still moving with the replication fork, but it's still making in the 5' to 3' direction. And so, again, this is showing the leading strand. So here's our double-stranded DNA. Here's our helicase unwinding. Here's our single-stranded binding proteins. Primase has already come in and added the primer. And then DNA polymerase is coming in, and again, it starts synthesizing this new piece of DNA continuously and towards the replication fork. At the same time, it's still adding at the 3' end of the molecule. Now, the lagging strand, like we said, it's going to be synthesized away from the replication fork and discontinuously, or not continuously. It's going to be made in these little fragments of DNA. And because we can only add to the 3' end of DNA. And so in the lagging strand, again, primase is going to come in, it's going to make a primer, then DNA polymerase 3 is going to come in, it's going to start synthesizing 5' to 3', and it'll do that until it runs into the next primer. And then at that point, it'll bounce backwards, to the new primer, the most recent primer, and then go down this way. And then if there's another primer, it'll do it, and it'll continue to do this all the way through the piece of DNA, making these little Okazaki fragments, which are these pieces of DNA in light blue. And then after you synthesize all of your DNA, both lagging strand and leading strand, well, on the leading strand, now you have all these chunks of DNA, but you need to remove these RNA pieces, right? Once we've done synthesizing DNA polymerase 1 is going to come in, remove the RNA primers, so literally cut them out, and then fill in the gaps behind with pieces of DNA. So helicase comes in, unwinds the double helix, single-stranded binding protein stabilizes single strand, topoisomerase prevents torque from occurring, primase comes in, puts a primer, DNA polymerase 3 comes in, replicates the DNA polymerase 1 comes in behind it, removes those RNA primers, and then fills in the gaps with pieces of DNA. Now the last thing that's going to happen in DNA replication, So we've removed all the primers and all we have is DNA now. But we still have these kind of chunks of DNA. They're not, held together by anything. So we're going to have another enzyme called DNA ligase is going to come in and ligate the two strands. If you ligate something, you tie them together or bring them together. And so that's what we're doing here. So DNA ligase will come in and make sure that these two pieces of DNA get attached to one another. And so after replication, you have two full strands of DNA. One of them started complete and continuous, while the other one started in fragments. But then eventually we glue those fragments back together with DNA ligase. And so here's a summary of all the enzymes that are used in DNA replication. And so helicase is going to come in, unwind it. Topoisomerase is going to prevent torque on the DNA molecule. Single-stranded binding proteins are going to stabilize the single strand. Primase is going to come in, make an RNA primer. DNA polymerase III is going to come in, land on the primer, and start synthesizing DNA in the 5' to 3' direction. And then DNA polymerase I is going to come in, cut out those RNA primers, replace them with DNA, and then finally ligase will come in and glue the Okazaki fragments together. Questions? Okay, so look, here's a summary of DNA replication again. Helicase unwinds the double helix. Single-stranded binding protein stabilizes the single strand. Topoisomerase prevents the torque. Primase puts in a primer. DNA polymerase III comes in, synthesizes. DNA polymerase I comes in, takes out the RNA primer, replaces it with DNA, and then you glue the pieces of DNA back together. Oh wait, there's more. I told you, I warned you, we're going to be going over this a lot. Okay, last time I promise. All right, y'all. So what's the first thing's going to happen? What's the first enzyme? Helicase, right? What does it do?
Unwinds the helix.
It unwinds the double helix. All right. What's the second thing that's going to come into play? Single-stranded binding proteins. They stabilize the single strand. This guy doesn't show topoisomerase, but he's going to be up here. What's topoisomerase going to do?
Relieve the torque.
Yeah. Do you know what I mean by relieve the torque? I feel like I didn't explain it very well.
Relieves the tension.
Yeah, it relieves the tension, because if you have something that's unwinding and unwinding, as you unwind it, the rest of it's going to kind of want to unwind as well. So like if you twist up two strings together, it's like, oh, this is perfect. Okay. You have kids, or you used to be a kid, and you sit on a tire swing or something, and you twist it and twist it and twist it. The minute you let go, it's going to, right? So helicase is doing the twisting and the untwisting. So topoisomerase is keeping it from all just unraveling at the same time. Okay. All right. Anyways, all right, so we've got the topoisomerase, we've gotten the helicase, we've gotten the single-stranded binding proteins. What's going to come in next? Primase. What's primase going to do?
It's the primer.
It's going to add these primers. And this picture is actually pretty nice because it shows you all these numbers are here are telling you they're labeling the Okazaki fragments. So this one got made first. There was an RNA primer right here at one point. This was the first primer that was made. The DNA polymerase came in, made this fragment, then jumped to the second one, made this fragment, jumped to the third one, made that fragment, so forth and so on. All right, so DNA polymerase III, what's its job?
To replicate the DNA.
It's making copies of the DNA. What's going to happen after DNA polymerase III synthesizes the DNA? What's next? What enzyme's next?
Isn't it the DNA polymerase I?
DNA polymerase I. What does DNA polymerase I do? It removes the RNA primers and then fills in the gaps with pieces of DNA. And then the last thing that's going to happen?
Ligase.
Ligase comes in. It's going to join those Okazaki fragments together. Okay, so that's DNA replication. Now remember, we talked about in the cell cycle that we have all these checkpoints, right? Checkpoints leading into the next stage. Well, one of these checkpoints is going to be right here. It's called the S checkpoint. It's not one that we talked about in detail, but its job is to regulate the passage of S into G2. And so at this checkpoint, what's happening is there's a lot of proofreading going on here, because we want to make sure that we didn't make any mistakes in replicating our DNA. Because a lot of times, the more we replicate our DNA, the more prone we are to errors. It's like, think of taking a copy of DNA, all right? And you're going to put it in a copy machine, and you're going to make a copy of DNA. And then that copy is going to be used to make another copy. And then that copy is going to be used to make another copy. So it's like making a copy of a copy of a copy. Eventually, it's going to lose some of its integrity, all right, because you're replicating it so many times. DNA polymerase is like, why am I here? Why am I doing this all the time? This is one way you get cancer, is when you do stuff that damages your tissues, you're damaging the cell. So what happens when you damage the cell? You have to make a new cell to replace it. What happens before you make a new cell? You have to replicate your DNA. So people who smoke, they're damaging their lung tissues. They're killing their cells. So their cells have to be remade to replace them. So every time you do something harmful to your body, you're going into the cell cycle. And again, you're making copies upon copies of DNA. So the more you're having to replace your cells, the more you're having to replicate your DNA. The more you replicate your DNA, the less effective it's going to be. But anyways, so that's why we have this checkpoint here. There's all these proofreading mechanisms. Make sure that we didn't make a mistake in DNA replication. And so a lot of these mechanisms, if they do come across a mistake, we have these DNA repair mechanisms that help, fix the issue. And so this is kind of like a proofreading, series of proteins that make sure that there's no mistakes in DNA replication. If they do come across a mistake,